Hi, how you doing? Doing okay. How are you? Well, doing great. Good. Okay, we're going to go ahead and get started. Welcome to our chapter meeting. I'm Hannah Priest. I'm the education board member, and I'm filling in for Victoria and Maeve because they're all at a conference. So bear with me if it's a little clunky, but I think we got it. We have a great speaker today. We got Gurney still did plant of the month, so we should be set to go. First, we're going to start off with our chapter business, and then we're going to have our featured speaker, Laurie Hamon. Chapter business, we had the Native Home Garden self-driving tour back in May. Thank you to all who participated in showing off their beautiful gardens, and also to those who came. And it was a really good time. Saw a lot of really fun home gardens, which is something that we don't usually do as a chapter. And it's good to see native plantings in progress and use at home. We also had our Yaqui Wildlife Center bus tour. That was super fun, a really unique opportunity to get access to the Yaqui Wildlife Center. I don't know if any of you guys have been, but I highly recommend going. There's also tours open to the public, but we were able to do a specific native plant tour where we hiked off the trail, saw some cool plants, including our lovely Carolina hedge nettle, which there are only 2 wild populations documented, and one is at Yaqui, and it was thankfully in bloom, and that was really fun. We also had our bumblebee survey at Lewis Ocean Bays over the weekend, and that was really fun. We had 2 sites that we surveyed. Oh. We had 2 sites that we surveyed at, and the first site, no bumblebee activity, but the second site was crazy busy with bumblebees. We caught 47 bumblebees in our 45-minute time, and it was really fun. I'm still working through the pictures, but here's a little sample. We have a little bee, and it's on our Carolina redroot, and they loved it. It was a really fun time to get up close and personal with bumblebees. And lastly, our chapter business, we have the state symposium coming up. So mark your calendars. It'll be September 18th through the 20th, and it's hosted by the Low Country chapter, and it'll be down at Fort Johnson in Charleston. General admission is $50, and that includes access to the symposium, field trips, like breakfast and coffee, snacks, and there will be additional add-ons for merch, evening socials, certain field trips. I don't know if you any of you joined us for our last symposium, but we had some really cool workshops that were a little extra, but all of our included field trips were really fun. So we're really looking forward to what the Low Country chapter has prepared for us. Okay, and I'm going to turn the time over to our featured speaker, Laurie Hamon. She is working with the North Carolina Natural Heritage Preserve and program, and she is a pollinator biologist, and she's going to speak to us today about pollinators. Let me see if I can switch screens. Here we go. Sorry, bear with me. Okay, Laurie, are you sharing your screen?
I am not yet, but I see that I now have the ability. So let me go ahead and do that.
Perfect.
Are you seeing my presentation?
I am not seeing that along the top.
Okay. Are you seeing my screen at all?
I am seeing you. Yes. Sorry, I don't normally do the Zoom stuff, so I'm.
No worries. Give me a sec. My state agency doesn't do Zoom, so sometimes there's a little bit of working things out. How about now?
There, I see it now. Okay.
Okay. And can you
Perfect.
see? You can see my first slide there?
Yes, let me switch my display really quick.
Let me know when you're good to go.
Okay, we should be good to go.
Cool. Okay. Well, I can't see any of y'all, so if you need me to repeat anything or need me to speak up, just speak up and interrupt me.
Yeah.
So when Hannah reached out to me, she mentioned a few different things that I've done over my career in recent years. So I'm essentially going to be giving kind of a whirlwind tour of some things that I've done in recent years related to plant and insect connections and conservation, especially from a pollinator perspective. And thank you so much for inviting me. So, um, so I thought that maybe to summarize some of the stuff that I'm going to be talking about, taking you through a tour of various projects I've been involved with. Some of the sort of overarching themes of this are that when we're studying insects, it's kind of hard to study insects, particularly pollinators, without knowing a little bit about plants. And if you are into plants, it's hard to understand them and get to know them without learning a little bit about bugs. I'm sure you've discovered that working in your gardens and seeing all the visitors your plants attract. And another theme from my talk is going to be the importance of community science, also called citizen science, for making advancements in plant insect conservation. And so I'm going to be walking you through four main things I've been involved with: a butterfly project, a Venus flytrap project, a bumblebee project, and then what I do currently, which is Natural Heritage Program in North Carolina. So first off, this was the very beginning of my career in plant pollinator interactions. When I was an undergrad at UNC Chapel Hill, I started working as a student researcher in the lab of Dr. Joel Kingsolver. And during that time, I started a research project looking at a massive data set of butterfly observations that were collected by community scientists throughout North Carolina since about the 90s, and using that data to make estimates, to make predictions about how butterflies have been faring under climate change in North Carolina, and which species are likely to be the most sensitive to climate change. And while I was working on this project, I became interested in how these data points were actually collected, the nuts and bolts of people actually going outside and recording a butterfly. And so as part of that, with Joel's support, we started in this lab something called the Mason Farm Butterfly Project, which was a community science project based at this reserve in Chapel Hill, North Carolina, where we would invite members of the public, do these public butterfly walks to actually centered around this one biological reserve in Chapel Hill to help understand how the butterflies at that site specifically were doing. And this site is managed by the North Carolina Botanical Garden, who had an express stake in understanding how their pollinators were doing and what clues that might provide about how their plants were doing. And there's a lovely little view of kind of a flooded bottomland forest in the reserve. And as part of this butterfly project, over the some 10 years that the project ran, we collected, and our volunteers collected, about 6,800 individual butterfly observations. That means sheer number of butterflies that were counted by at least 50 community scientists, probably a lot more, because sometimes people would just say, I'm here with my group of friends counting butterflies, and they wouldn't elaborate how many of them there were, representing roughly 54 species. And it was a great experience. It was co-coordinated by myself, and then there's a picture of our co-coordinators, Katie Moore and Elizabeth Moore. And it was so lovely to meet so many enthusiastic people trying to learn how to ID butterflies and what insights those butterflies can provide about habitat health. The Mason Farm Butterfly Project is officially shuttered. Maybe that's a bit too dramatic a word. It's concluded. It's not being actively run anymore, but I will mention that Jeff Pippin, I'm not sure if anyone is familiar with him, but he's a local butterfly expert, and he has started a Carolinas wide, both North and South Carolina, butterfly monitoring program that essentially uses the same protocols. And in this program, you can adopt a specific route, check it, ideally weekly. I probably honestly do it about once a month. And record the butterflies you see there and help provide information about butterfly health and the environments that they live in. And there's a picture from my assigned route, which is in Chatham County, not at Mason Farm Biological Reserve. But I mention this because this is a way that if you're interested in butterflies, you can get involved still in this project, adopt a route, and really get to know it and the butterflies there. So that was sort of how I got my start. And so switching gears a little bit, the butterflies were undergraduate, but for grad school, I knew that I was interested in plant pollinator interactions from a conservation perspective. And I just happened to fall into this research group at NC State who wanted to take on a grad student who were studying Venus flytrap, which I said, of course, I would love to study the pollinators of Venus flytrap. And so those are the primary investigators there, the people who I answered to, Dr. Clyde Sorensen, Dr. Becky Irwin, and Dr. Elsa Youngstead at NC State University, and a couple of the folks that help support me are listed up there at the top when I was doing this project for my PhD. And so Venus flytrap as you may very well know, and probably got a chance to see them recently, it sounds like, are a rare species. They only occur natively in Southeast North Carolina and Northeast South Carolina. And the actual identity of the bugs that pollinate the flowers was only recently described in 2018 by this research group. And they found that mostly sweat bees and a colorful group of beetles are probably the major players in terms of pollinating Venus flytrap, getting pollen from Anthrodistigma and allowing it to produce seeds. And another interesting aspect that was known when I came in on this was that if you remember that first picture, Venus flytraps keep their flowers maybe about a foot above the actual traps themselves, and they are very good at not eating their own pollinators. So here, this little diagram is showing up top the suite of insects that visit the flowers, and up the bottom, the suite of insects that get caught in the traps. And then it's highlighting the very, very small overlap of species or types of bugs that appear both at the flowers and caught in the traps. And it's mostly not very important pollinators getting caught in the traps. So the things that get caught in the traps are mostly things that crawl along the ground, like ants and spiders. So my PI would always say that it should be called the Carolina spider trap rather than the fly trap. And it's hard to distill in a 20-minute presentation or a sub 20-minute presentation sort of the main findings of my research, but I would say the key takeaways are that insects are important for Venus flytrap reproduction, both as a source of nutrients and as pollinators. They're visiting the trap, visiting the flowers. So I just quickly summarize, I think, one of the more fun studies I did, which is I was interested in how important getting insect prey is versus getting pollen from pollinators for reproduction in Venus flytraps. So for this study, I had a bunch of Venus flytraps in the field, in the wild. I gave some of them prey in the form of frozen crickets. So I put the frozen crickets in the traps and then gently massaged the traps until they decided it was indeed food and decided to digest it. And then there were a few I supplemented pollen to the flowers to represent sort of the best case scenario version of pollination. And then there were some lucky individuals which got both prey and pollen supplemented to them. And one of the more interesting findings that I got from this was that so I fed some plants in 2020, and then a few months later, those plants produced significantly. It looks like a small number, but it was significant considering each flytrap produces about 11 flowers per individual. Those fed individuals produced an average of 1.9 more flowers per individual than the ones that weren't given extra food. And overall, it mathed out to those fed plants producing more seeds compared to the unfed plants, which makes intuitive sense. If you get more nutrients, maybe you can support more babies. But it was still really interesting to see. Another aspect of my graduate school research connects to what I currently do, because I was more or less headhunted by the Natural Heritage Program, who had been asked by the U.S. Fish and Wildlife Service to do a status survey for how many Venus flytrap populations were in the wild and how many individuals there were and how Venus flytrap was faring. At the time, Venus flytrap had been submitted as a candidate for listing under the Endangered Species Act, and it was still a little unclear how many populations of Venus flytrap there were and how many individuals there were. At the time I started the survey, or heading up the survey, working part-time for Natural Heritage Program and part-time as a grad student at NC State, there was an estimate of a maximum of 158,000 Venus flytraps known in the wild. But the survey revealed that there are at least about 180,000 Venus flytraps in the wild, probably close to a million, accounting for underestimates from our survey efforts. So this was a huge multi-agency collaborative effort that took place over North and South Carolina. And there's Dan Hannon and some of our interns or our field assistants in the bottom. And it was just a real privilege to help head up this survey effort. And so 1 million fly traps sounds like, okay, so fly traps are safe, they're doing well. But I think it's worth pointing out that from a conservation perspective, there are a lot of individuals, but those individuals of fly traps are concentrated in just a handful of mega populations. So of the 880 odd thousand, 100,000 Venus flytraps that are in the wild, about 67% of them, like 2/3, are in just 3 populations, which isn't ideal. You would love lots of healthy populations so that if one blinks out or the land management changes at another site, you can ensure that you still have healthy populations. So that was an interesting finding, is that sure, there are a lot of fly traps, but they're concentrated in just a few locations. And this graph is showing our A, B, C, and D ranked populations. An A ranked population has more than 2,000 individuals in it, and 98% of them are just in a handful of A ranked populations, and 67% of them are in just 3 populations. And another interesting aspect of this status survey that I got to help out with was the Fish and Wildlife Service was also interested in the status of Venus flytrap cutworm, which, if you're not familiar, Venus flytrap cutworm is a rare moth that depends almost exclusively on Venus flytrap as a host plant. So Venus flytrap is rare, and this moth is way rarer because it depends on a rare plant for food. So I got to do some surveying for this species, and really fell in love with this species as like a case study of rare plant, rare bug, and how tightly they are associated. And remember this guy because he's going to come up later. Okay, so that was grad school. After grad school, I did a postdoc, and then after my postdoc, I got involved working for this nonprofit called the Xerces Society for Invertebrate Conservation as their local coordinator for the Southeast Bumblebee Atlas. And it sounds like potentially y'all have already participated in some surveys for these, or maybe you're going to be doing some later. But it was a really thrilling project to work on. Basically, the goal of these projects are to understand how bumblebee populations are faring across the United States. We have some 40 odd species in the United States. In the Southeast, we have maybe 15, and about half of them are considered of conservation concern. Maybe they're declining or we haven't seen them for a number of years. Bumblebees are especially effective pollinators. They're generalists. They visit a bunch of different plants. They're big and fuzzy and literally bumbly. They're great pollinators on a bunch of different things. And so it's important from conservation and agricultural perspective to understand how well they're doing. And so the goal of this project was to get community scientists involved, actually asking them to get out, capture bumblebees, record what plants these bumblebees are visiting, put them on ice to slow down their movements so we can take good voucher quality photos of those bumblebees so that an expert at the Xerces Society, in this case it was me for a couple of years, can verify what species people actually saw. And on the left is the map of the survey grid cells as of 2026, I believe. And so the way similar to the butterfly survey, it asks people to adopt an area and then commit to surveying it over a particular time period. And after I left in 2024, Avery Young took over for me, heading up the project. And then on the left is the number of surveys that have happened for this project as of 2026. And it's been thrilling. I now have participated as a participant rather than a coordinator to see this map fill up. Avery, unfortunately, finished her role at the Xerces Society earlier this year because she went to go work for the Natural Heritage Program. So she and I had the same sort of career trajectory. We went from the Bumblebee Atlas to the Natural Heritage Program. So this is where I currently work as a pollinator biologist, and it has been a really exciting chance to tie together a lot of the themes and things I'd already been thinking about for about a decade about rare plants, the pollinators that depend on those rare plants, the plants that those pollinators depend on. So what the North Carolina Natural Heritage Program does is we track the locations and statuses of rare species and natural communities throughout the state of North Carolina. We delineate which natural areas are high quality for the purposes of land acquisition and conservation planning. And we dedicate certain areas as nature preserves and heritage areas and work with landowners to actually manage those to make sure that those rare species and natural communities will stay on in perpetuity. My role is specific to the bugs. So my title is pollinator biologist, and indeed I do focus on the bees and butterflies, but it covers bugs generally, and it's been really exciting to learn about groups I heretofore didn't have much experience with. But just to get an idea of what sort of insects we're interested in North Carolina, on the left, far left, are some of the kinds of bugs that we track at Natural Heritage Program. The middle is the number of species. Apologies if you heard my cat there. The number of species that we actively track at Natural Heritage Program. And on the right are what we call our watch list species, which are species which are likely to become significantly rare in the future. We're sort of keeping an eye on them. So this is just to give you an idea of how the numbers shake down. So the butterflies is interesting. In North Carolina, we have maybe 170, 180 odd species of butterfly known from the state, and then 38 of them we track as significantly rare. And just an example of one of the species we track, which is really tightly associated with a specific plant, we track Andrena rudbeckiae, which you might be able to tell from the name is a kind of bee that specializes on Rudbeckia. So it's hard to think about bugs without thinking about the plants at the same time. And so what we do at an agency to actually track these species is we do it a number of different ways. I feel very privileged that about 20% of my time is spent doing direct surveys where I'm actually going out to sites where a rare bug is known or suspected and doing actual direct surveys on the ground. And we're very lucky we got the chance to hire 2 new invertebrate entomologists for a mountain-specific project. And there they are on the left, assisting with a snail survey in Catawba County in North Carolina, one of whom is our new terrestrial invertebrate entomologist, Avery Young. So NHP has poached 2 people from Xerces, but there's no hard feelings. So that's about 20% of my time. And then 80% of my time is actually wrangling data that other people have gathered. And so community science is a really big part of what I do. Things that people submit to iNaturalist or submit to me directly through our online forums end up informing management on these managed areas that we help our landowners manage. And so a rare species seen in one location ends up in our system, and then we can help protect that specific parcel of land to protect that species. And that screenshot there on the right is we've recently started our own official iNaturalist project specific to arthropods, so that's insects and crayfish records that occur in North Carolina. So if you go up into the North Carolina border and you see something rare, feel free to join our INAT project. It could really shed some light on the conservation needs of where it was seen. And so I've sort of jumped around for a number of different projects, but hopefully they've given you some ideas of places to get involved, thinking about plant-insect interactions. It's very, very hard to think about one without thinking of the other. They're very tightly connected. anyone making observations, whether it's in their back garden or, one they're on a hike, those observations can help contribute to making conservation decisions. So whether you adopt a butterfly route or you do the bumblebee survey or you submit some sightings to the South Carolina Natural Heritage Program, that's all really essential data. And on the picture on the screen is a Venus flytrap cutworm moth that I saw back in May. I am very, very happy that in my current role, I get to keep looking for this special little worm that feeds on Venus flytrap. And this sighting represented a new population for this species. So before there were only 3 known populations for the Venus flytrap cutworm. This was a new location, so it bumped it from 3 to 4 known locations. And I'm just very, very fond of this little worm, and I'm glad I've gotten to study it for a number of years now. So my contact info is on the screen. If you see something cool, feel free to reach out. I'm happy to take a look at it. And with that, hopefully there's some time for questions.
Thank you so much for your presentation. That was fantastic. Just lots of clapping. Does anyone have any questions? Anyone in the chat? Yes, in the back.
So, we have really good space right down the road here. And I know you did a survey out there for Venus flytrap, but the number that you arrived at for the preserve keeps, it's like urban mythology. Every time I hear somebody say the number, it changes. So what was the actual count of Venus flytraps on Ocean Bay?
Would you mind repeating that? Someone closer to the mic.
I got you.
So Lewis Ocean Bay is right down the road here. And the number of Venus flytraps in that preserve, it varies all over the place depending on who says the number. And so how many did you actually count there?
It's been a number of years since the actual survey. It went over the 2,000 mark. And then I think someone actually came around after me and found a couple thousand more. So if I had to ballpark it, it's somewhere in the range of maybe 5,000, 6,000 individuals. But I'd be interested to know what the most recent number someone told you was.
The most recent number I heard was 30,000.
I think that's a bit high, but I could be proven wrong.
Okay. Thank you.
Any other questions? Yes. Run.
Are carpenter bees the same as bumblebees?
That's a great question. They're related. They're in the same family, but carpenter bees are a different genus. And they have an entirely different lifestyle. So they're somewhat related, but they are very different bugs in many ways.
Also common. I just posted, I don't know, a dozen pictures of different pollinators on my mountain mint in my backyard. And the challenge that I'm having is getting them to sit still long enough to get a good picture. So what's the process for capturing them and getting them on ice without injury to them or me?
Yeah, that's a great question. Thankfully, bumblebees that are engaged with collecting pollen are very disinterested in actually hurting anyone. I'm casting about, here we go. So for the bumblebee atlas, the way we do it is we catch them in a net, or if they're preoccupied with mountain mint, you could probably just do this directly. We'd use vials like this. Some people use medicine bottles that they've washed out really well, or small mason jars. You can catch the bee in there, put it on ice, like put this directly on ice for about 20 minutes. And after that point, she'll be chill enough that you can take pictures of her from multiple angles. And then after about 5 minutes, she'll warm up and fly away. And you can do this strategy for a bunch of different bugs. Flies tend to not chill out very well. They wake up pretty soon after they start warming up. But yeah, you can try it for a number of different things to help get good photos.
Hey, I just had a question. Did your research for fly traps take you onto much in individuals' private lands around the Carolinas, or just like Lewis Ocean Bay public lands kind of situation?
Yeah, so the survey started in 2019, and we were really focused on public lands. Every once in a while, a private landowner we would connect with them, and they would let us on our lands, though that didn't add to the number much. And then in 2020, the focus was private lands. But as you can imagine, in 2020, it was really hard to get a good response from private landowners for getting on their lands. So, so that count does include some private lands, but I wish it included more. And part of that is the just response rate of landowners we would reach out to.
Okay, any other questions? And I don't see any questions in the chat, so I thank you so much for your time, Laurie, and for speaking to us. That was really awesome. I hope that it drums up a lot of interest in our chapter Bumblebee surveys, and it was great to hear about all the work that you've done.
Awesome. Thank you so much, everyone.
Get my screen back up. Okay, I'm not sharing this. Hold on. There we go. Okay. We have plant of the month. I just got this set up and now I have to go to a different one. Here we go. Plant of the month. And our lovely Tyranny, even though she is not here with us physically, has manifested herself spiritually to share with us the plant of the month.
Well, not actually here. I'm at co-leaded plant conference this week, but I couldn't leave you without the much awaited plant of the month. The July 2026 The July 2026 plant of the month is one of my favorites, Ticreum canadensis, commonly known as American or common germander, or as May's husband Davey likes to call it, American Charmander. A little Pokemon reference for millennials in the group. Common germander is an herbaceous rhizomatous perennial in the Lamiaceae or the mint family. It spreads via underground stems, but not as aggressively as other members of the family. Common germander grows best in part sun to part shade and moist sandy loam to sandy clay soil. The bitter chemicals in the leaves make this plant resistant to herbivores like rabbits and deer. Common germander is exactly that, common. It can be found growing in rich bottomland, prairies, marshes, from Nova Scotia, south to Florida, west to Texas, Oklahoma, and Mexico. While each pinkish purple flower is on the small side, they make up for their individual size by growing in clusters on spikes that are 4 to 5 inches tall to make a beautiful floral display in mid-July. Like all plants in the mint family, common germander has square stems. The leaves are opposite and simple with serrated margins. The stems and leaves are covered in lots of tiny hairs, or otherwise known as pubescent. Small seeds of common germander require 60 days of cold moisture adaptation to germinate. But plants are easily propagated by dividing root cuttings in spring or fall. Common germander is a fantastic plant if you want to support pollinators in midsummer when many of the other garden staples are done for the season, and we haven't yet hit after goldenrod season. A wide variety of bees, butterflies, and moths are attracted to the flowers. Hummingbirds will even visit the tube-like flowers on occasion.
I think that's about, wait, no. Forgot this slide has 2.
So if you haven't thought you can let this amazing plant spread, I highly recommend adding it to your garden. See you all in September.
Thank you, Tierney. She did not. Let me see if. Yeah, it's gonna be very hard to hear. Oh dear. The growing conditions, I would say, I'm hoping that Tierney comes and pipes up, but I would say it's in the mint family. It's going to be super highly adaptable to most conditions.
We have a patch over at the park that we would probably see if you come out for the CP Cameron survey, right at the North Beach access left. And it's growing practically. So, yeah. I mean, it's right on the edge of the shrubbery, but it's mostly summer.
Tierney's here. She's watching. She might pop in. Waiting. Hold it for a second. No, they're watching through the same account that I'm using. So if she answers, it'll be in the chat. We'll circle back. Oh, there we go. Medium to moist, sandy loam to sandy clay soils, and part sun to part shade. So very highly adaptable. Okay. Upcoming events. We have next week, Tuesday, we have our chapter is hosting the Myrtle Beach Green Drinks, and it will be at the Grand Strand Brewing Co. So if you guys are interested in coming, we'll be there. We'll have trivia. There'll be drinks, and it'll be a good, fun social. We also, this next Saturday, we have, not tomorrow, or 2 days from now, but the Saturday after that, we have the Sea Beach Amaranth monitoring. And that's going to be at 8:30 a.m. to 11:30 a.m. at Huntington Beach State Park. That's a really fun time, looking and cataloging all the new growth on the Sea Beach Amaranth. Mike, would you like to pipe in?
Yeah, so,
I got this for you.
we're specifically going to be looking for naturally recruited plants. So most of y'all are probably familiar that we've worked with Brook Green Gardens for a number of years, trying to reestablish the plant. So any of the ones that were planted from Brook Green, those are cordoned off. So we're not counting those, but we're specifically looking for the naturally recruited plants. And last summer we hit a record of over 500. So. Now, with the drought conditions, I'm not sure we're going to get that, but and I've been making a point to not peak. So I am going to, anytime I go in an area where there might be some, like, I'm not going to look, I got to save it.
Okay. We also are going to be having a composting workshop on August 1st. We're going to have Maureen Mulligan from Georgetown County come up and teach composting, and we should have a little take-home activity that we're going to be doing. Location, I still need to schedule. I just confirmed with her this week that she was going to be able to come up on August 1st, but that class is going to be 10 a.m. to 11:30 a.m. and I'm going to try and schedule it here to make it easy for everyone. Regardless, it will be in the Conway vicinity if this room is unavailable. And we are also trying to partner with Sustain Coastal for that. So hopefully I hear back from them. But mark that on your calendars. We have another Bumblebee survey, and that will be at Huntington Beach State Park on August 15th, 8:30 a.m. to 11 a.m. And we're going to be meeting at the Old Entrance Road. If any of you guys have done the invasive species removals, it's a little bit further along that road. And that should be a good time. We found, Mike and I went out last Friday to check possible locations, and we think we found a good one. So it should be a good time. We even saw some bumblebees, so they're there. We are also going to be having a mothing event at Huntington Beach State Park, and that's going to be September 4th, 9 p.m. to 11 p.m. It's going to be in that same bumblebee area, Huntington Beach State Park, the Old Entrance Road. And that's going to be a really fun event. We're going to be hanging up a white sheet, shining light on it, and seeing what types of nocturnal insects come. Because, fun fact that I just learned, 60% of insects are nocturnal. And nighttime activity, insect activity, is up to a third higher than daytime insect activity. So this is a good chance to get to know all of our cool nighttime insects. Hopefully we'll see a lot of fun moths. And here's a lovely picture of a Luna moth to get you all interested.
One thing I do want to point out, I did not think.
Yeah, you did.
One thing I do want to quickly point out, please do not be late, because the park gates close at 9. They, in the past, they've always closed at 10, and that's kind of what was in my mind when we scheduled this. So as long as you're there, I would say get there like 8:45,
I'll change it to 8:30.
8:30. That even better.
Yeah, we'll get everyone in. Okay, 8:30 to 11.
And I will make sure everybody gets that.
We won't be locked in there overnight. September, we also have another bumblebee survey. We're going to be going back for our second survey at Lewis Ocean Bay. That's going to be September 12th, 8:30 a.m. to 11 a.m. Rain day, September 13th, if we have bad weather. That should be a good time. We have two locations that we're going to be, two survey spots that we're going to be surveying. One at the first fork of the road and one down the edge of one of the Carolina bays. So should be really fun. Come join us, learn about our native bumblebees. Our survey last week found all 5 of the normal species that we find here, which was good. None of the other surveys we've done in the area have had all 5, but this last one did. So hopefully we'll have good bumblebee activity again in September. Our next chapter meeting is going to be September 17th, same time, same place. And we're going to be having speakers from the South Carolina Heritage Trust Program. So kind of the sister organization to Laurie's organization, the North Carolina Heritage Program. They're going to be joining via Zoom and talking about some of the work that they're doing here. So that should be fun. And a reminder, the very next day is going to be the state symposium. So mark that on your calendars. That'll be that next weekend. Hopefully we'll all see you there. I'll be there. It'll be fun. Okay, do we have any member updates for the mic? Anybody? Yes, the question was, are all the events on the SCNPS website? And they are. I added them 2 nights ago. So they're there. They're on the calendar. It's official. Anyone else? Questions, updates? Yes. Oh, fantastic. We have seeds to share. So sorry for our members who are Zoom. We cannot share these virtually. Anyone else? Updates, comments, questions, concerns? Telling me I did a good job. There we go. Thank you. Okay. So that is it for our meeting today. No more questions. Oh, good job from the crew at Cullowhee. So thank you all for coming and for your patience with me fumbling through Zoom. And we hope to see you next time and at the symposium. Symposium will be fun. Okay. Have a great night.
Nominated cucumbers in my garden.